Moving forward with our Phase 1 project, I will be continuing the TA cloning procedure. First, I carried out transformation, where competent cells take in external DNA as part of their own. Using plasmids with our insert, we're able to use these cells as little factories to produce many copies of our sequence of interest. Since we're using blue-white selection, colonies that are blue do not have our plasmid, while white colonies do. This visualization lets us select colonies that have our plasmid and insert to then culture overnight. On Wednesday, I came into the lab to start an overnight culture with the white colonies from the plates prepared on Monday. The white colonies are the ones with our plasmid and insert. This process went smoothly as I had ample amounts of white colonies to select from. For my overnight cultures, I picked 4 colonies from the 100 plus microliter plate. It's important to note that I used a 200 microliter pipette instead of a sterile loop to collect the colonies. I dipped the pipette tip in the single white colony and dropped the entire tip into the tube with LB plus amp. The pipette tip will be removed on Thursday. Since there was no more room on the individual tube holder in the 37-degree shaking incubator, I used a glass beaker and tissues to hold our overnight cultures. On Thursday, I prepared a single plate that contained all 4 of these overnight cultures. The purpose of preparing a new plate is that the resulting colonies will have only our sequence of interest, since I used blue-white screening in the previous lab session. Using the aseptic technique, I plated these 4 colonies. Following the NEB Monarch Mini Prep protocol for plasmid purification, I isolated the DNA from the host cells. These cells served as factories to produce multiple copies of the sequence of interest. However, once completing that step, they are no longer necessary and are lysed and discarded in a pellet. After being treated with different wash buffers, the DNA gets stuck within the spin column and is eluted with nuclease-free water. I used the nanodrop instrument to analyze all 4 samples and found that they all had strong DNA presence with nanodrop curve peaks around 260 nanometers and high concentrations of 150 plus nanograms per microliter. All 260-280 ratios were within the parameters outlined by GeneWiz, which is 1.8 to 2.0.